So we had a plate, and we had patients 1 through 4. We had columns A, B, and RH. So I'm just going to look at one of them here. I'll try just patient 1 briefly. So we just put 1 drop of patient 1's blood in each of those 3 wells. Okay. Now I added 1 drop of the appropriate antibody in each well. Remember, antibodies cause clotting in antigens they're specific for. So antibodies to A will cause clotting in any blood that has A antigens. Antibodies to B will cause clotting in any blood that has B antigens. Antibodies to RH will cause clotting in any positive blood. So with our patient, we've got simulated anti-A serum. So this is going to clot any blood that's got A antibodies. So put 1 drop of the A antibodies in this person's blood. And B, drop B antibodies in the blood. RH, and put 1 drop of RH antibodies. Probably not going to have enough time to see some actual results for this one. Like I said, I've got a whole other video about blood typing. But in the end, we're going to expect to see, for our first patient, we're going to see clotting in this well only. There's not clotting in this one. There's not going to be clotting in this one. So if you have clotting in this well only, that means only the B antibodies clotted. So that means we have only B antigens present. If there's only B antigens present, it means, of course, we have type B blood. Now, we have to decide if it's positive or negative. Remember, there's no clotting in the RH. So if there's no clotting in RH, it means there's no RH antigens present. So it means the blood is minus, or negative, rather. So this person's blood type is B negative because the only antibodies that clot are the B antibodies. We don't have RH clotting or A clotting. Okay. So I want you to also be able to take a look at one of these blood plates on your final exam and be able to tell me what person's blood type is. It's a very important skill to be able to do for your final exam. And the next thing we looked at was paternity problems. So also I would expect you to be able to, knowing people's blood phenotypes, be able to decide what kind of kids they could have, what blood types of the kids they may have, or look at someone's blood type and decide if he could really be the father of a person or not, or she could be the father, or rather the mother of a person or not. And expect to be able to work the paternity problems just like you have in your manual. If you're not sure about how to do paternity problems, I've got another video where I work several paternity problems out. Okay. Final thing we looked at was multifactorial inheritance. So let's walk over to the chalkboard while I'll talk about it. Okay, in multifactorial inheritance, there are traits in which you have lots of different genes involved. So many traits that exhibit a wide variety in the trait are multifactorial. For example, human skin color is multifactorial, and also human height is multifactorial. So we worked a problem and we assumed that there were only 4 genes for height. And it's also important to keep in mind that in multifactorial traits, environment can be a factor too. So you may have genes to be really tall. If you don't get a lot of protein when you're younger, you may grow up to be fairly short. Because multifactorial is something that sometimes can be influenced by environment as well as just your genes. So like I said, we assume we had 4 genes for height. And that means we're assuming that we have 8 alleles for height. For example, we just called them A, B, C, and D. Okay? Now, in multifactorial inheritance, we don't call alleles dominant. We don't call them recessive. Instead, we call them contributive or non-contributive. A contributive allele will increase a particular trait, and non-contributive allele will just have no effect on it at all. So, let's take a look at some examples. We wrote out contributive alleles with uppercase letters, non-contributive ones with lowercase letters. So our first individual here has A, B, C, and D. You can see he has only contributive alleles. That means this person is going to be very tall. And this person has no contributive alleles at all, so this person is going to be very short. Now it's important to remember this person is not going to be 0 inches tall. we're going to assume there's a particular base height that people are, that someone with no contributing alleles will be. For our example, we assumed it would be 60 inches tall. So we'll assume this person is 60 inches tall. And this person here, you can see this person has half contributive alleles, half non-contributive alleles. So this will be a person of average height. This person down here is also a person of average height. Half non-contributive alleles, half contributive alleles. So the whole point of this exercise was we were trying to decide how many inches of height each contributive allele would add to a person's height. So right off the bat, we know that someone with no contributive alleles will be 60 inches tall, because that's their base height. That will be this person. But in order to decide how many inches over the base height a particular contributive allele added, we first had to find the average height of people in our classroom. So that's what we did. We'll walk over here. So we got our classroom, and we found out that the average height of people in the class was 68.6 inches. Okay. Now, someone who is of average height has 4 contributive alleles, 4 non-contributive alleles, right? So we applied a formula. We took our average height, we subtracted the base height of 60 inches, we divided by 4. Remember, 4 is the number of contributing alleles for someone of average height. So 4 out of 8 alleles contribute. We found out that we got a value of 2.1 inches. That means that every contributing allele that you have will add 2.1 inches to your height over base height. So that was probably worked out with multifactorial inheritance. Remember that it's not only genes that's a factor, it's also environment. And there's a lot of stuff to study in this chapter, so make sure you learn the terms. Also, make sure you know how to do very simple Punnett squares. We have a video about how to do that. And make sure you can write the genotypic ratios and phenotypic ratios of crosses. I also would like for you to be able to know our examples of simple dominant recessive traits that people in the class may have had. Know about morbidity genes and sex-influenced genes like pattern baldness. And I'd like for you also to be able to do blood typing and work paternity problems